You're once again welcome to Muhumuza Naphtali medicalscience.com, and I always remind you to subscribe. What is the importance of subscription? Whenever we release videos, you are able to be notified whenever you subscribe. So make sure you always subscribe to always be updated whenever we release a video. So let us start. So today we are looking at digestion, absorption, and metabolism of carbohydrates. We want to see how does, when we eat our food, how is it digested in our body, that is an alimentary canal, and how is it absorbed into the blood? And once it reaches in the blood, how does the body utilize it to provide energy? So let us begin with digestion. Digestion of carbohydrates, it starts in the mouth. We see the events occurring in the mouth, then in the stomach, in the duodenum, and small in the duodenum and ileum. Whereby in the mouth, whenever you take food containing carbohydrates, that is majorly starch, in the mouth we have two types of digestion occurring. That is the physical digestion. Then we also have chemical digestion. Whereby physical is by mastication. That is action of teeth. Mastication is action of teeth, whereby during the process of chewing, the teeth breaks down these carbohydrates into smaller portions, which can easily be swallowed. Then, as the role of the tongue is to mix, as it is mixing, we see production of saliva. And this saliva contains liquid like water, which moistens the food. Then we see production of salivary amylase enzyme. And this salivary amylase is the one that is responsible for chemical digestion of carbohydrates in the mouth, whereby it hydrolyzes starch to maltose. Maltose plus dextrins. This is in the presence of salivary amylase and water. So salivary amylase catalyzes the hydrolysis of starch in mouth into maltose and the dextrins. So this is the only digestion that occurs in the mouth. So once food is formed into a bolus, then it is swallowed through the esophagus into the stomach. And within the stomach, because in the stomach we have the production of gastric juice, and this gastric juice containing hydrochloric acid, which is an acidic which renders acidic pH. This one, that's why we see there is less digestion of carbohydrates in the stomach because of presence of acidic pH, which inhibits action of amylase enzyme. So there is no digestion of carbohydrates in the stomach. Though some books, when you're reading some books like Anatomy and Physiology by Totora, they will tell you that there is some enzyme produced in the mouth called the gastric amylase, but not so much common. So that's why in summary, we say no digestion. No digestion of carbohydrates in stomach, no digestion in stomach, due to presence of acidic pH that inhibits the action of salivary amylase. So when the food chyme in the stomach reaches, so in the duodenum we have the food chyme. The food chyme in the duodenum, it is going to trigger the production of pancreatic juice, and this pancreatic juice from the acinar cells of the pancreas, this pancreatic juice contains an important enzyme known as pancreatic amylase. And this pancreatic amylase it catalyzes the hydrolysis of starch, the remaining starch, which remained because some of us we eat very faster. And before salivary amylase converts starch to maltose, we have already solved. So the remaining starch in the duodenum is hydrolyzed to maltose by pancreatic amylase. Catalyzes the hydrolysis. Then the dextrins which were formed in the mouth, they are converted still by the same enzyme. We have the dextrins being hydrolyzed by pancreatic amylase to alpha limit dextrins. Alpha limit dextrins. So this is the only digestion occurring in the duodenum, whereby presence of food chyme triggers the production of pancreatic juice, which contains pancreatic amylase, which hydrolyzes starch to maltose and dextrins to alpha limit dextrins. From there, the food continues into the ileum, where in the ileum there is a final digestion, where final digestion of carbohydrates take place. So final digestion occurs in the ileum, which is the last part of the small intestine. And we know that in the ileum, there is presence of food in the ileum, food chyme in the ileum triggers production of succus entericus, which we locally know as intestinal juice. You can call it intestinal juice. And this intestinal juice contains enzymes for final digestion, contain enzyme like sucrase, enzyme like maltase, contain enzyme like lactase, and contain another enzyme called dextrinase. Dextrinase. So it contains 4 enzymes which participate in the final digestion of carbohydrates. For example, when the food contains maltose, when the food contains lactose, when the food contains sucrose, when the food contains alpha limit dextrins, we see maltose and all of them are hydrolysis reactions, so there is presence of water in each. All of them occur in the presence of water, whereby the water breaks the alpha-1,4 glycosidic bond, whereby maltose is hydrolyzed by maltase to glucose plus glucose. Then lactose is hydrolyzed by lactase enzyme to glucose plus galactose. Then this one, sucrose, is catalyzed by sucrase to glucose plus fructose. Then alpha limit dextrin is catalyzed by dextrinase enzyme to form glucose. So this one marks the end of digestion of carbohydrates, whereby we have seen the events that occur in the mouth, where we have both physical and chemical digestion. Physical by teeth, then chemical by salivary amylase, which is hydrolyzing starch into maltose and dextrins. Then in the stomach, there is no digestion due to presence of hydrochloric acid. Then as food continues into the duodenum, there is production of pancreatic juice. And this pancreatic juice contains enzyme pancreatic amylase, which is able to hydrolyze starch and dextrins to maltose and alpha limit dextrins, respectively. Then as it continues in the last end of small intestines, we have production of succus entericus, which contain 4 enzymes which perform the role of hydrolyzing these disaccharides into simple sugars. Then from here, this one marks the end of digestion. Now we can talk about absorption. Absorption. How are carbohydrates? How are these simple sugars like glucose, galactose, fructose, how are they absorbed into the bloodstream? We know that in the ileum we have intestinal wall. If this is the intestine and this is the rumen of the small of the ileum, and we have glucose inside the rumen. Glucose, there are majorly two ways of absorption. One process is known as active transport. They can either be transported by active transport or it can be by facilitated diffusion. Facilitated diffusion. Whereby under active transport, it requires energy in form of ATP because means in the rumen there is little digestion compared in blood. If this is blood, meaning blood has higher, so you are moving from low to higher. So we need energy. So it is against concentration gradient. Against the concentration gradient. That's why we need energy to move glucose from region of lower to region of higher. Then under facilitated diffusion, this one, it is whereby glucose is higher. If I have glucose here, glucose is higher in the rumen than in blood. So what happens, we are moving along the concentration gradient. So this is along the concentration gradient. So no energy is needed, no ATP is needed, no energy, because it's along the concentration gradient. And how does active transport occur is that I have a transporter in the rumen of the small intestine, the intestinal wall, whereby when sodium is entering inside the blood, then glucose jumps on his back and they move together into blood. So glucose also is transported into blood by the process known by the transporter known as sodium dependent glucose transporter. Which involves the hydrolysis of ATP to ADP plus phosphate inorganic. We normally abbreviate it as C, sodium dependent glucose transporters. Those are the ones that enable active. So active transport against the concentration gradient is by sodium dependent glucose transporters. Then, of course, along this is facilitated diffusion, whereby glucose moves freely along the concentration gradient. Then you can ask yourself, how is galactose and fructose absorbed? Sometimes galactose and fructose, they are first converted to glucose. You remember we have an enzyme called epimerase. And epimerase can easily convert galactose to glucose so that it can easily be absorbed via the intestinal wall into the blood saturation. And once glucose is in blood saturation, we have glucose in blood saturation, it is taken to it is transported via hepatic portal vein, and it is taken to the liver. And in the liver, that's where metabolism takes place. So whenever you're asked to discuss absorption of glucose, you tell us how it crosses the intestinal wall into blood, and how is it transported from blood to the liver. That is the end of absorption. And majorly it is two ways: active transport, then facilitated diffusion. I have not told you about facilitated diffusion. Facilitated diffusion is like gates, that this wall has gates. It has what? Gates. These gates are called GRUT transporters. And especially in the intestines, we have GRUT 5, because we have glucose transporter 1 up to 5. So these glucose transporters, whenever glucose is high in rumen than the one in the blood, These gates are always closed when there is not much glucose. But once glucose goes high, they open and allow the glucose to move into the blood. So simple, the facilitated diffusion is by glucose transporters, what we abbreviate as GLUT transporters. In summary, we are saying active transport. Active transport is against the concentration gradient facilitated by sodium-dependent glucose transporter. Facilitated diffusion is along the concentration gradient, so no ATP is required. And it is facilitated by GLUT transporters, or what we call glucose transporters. And once in blood, we have seen that glucose, after being absorbed in blood, it is transported to the liver via hepatic portal vein. And when inside the liver, that's where we're going to see how does the body deal with this glucose that has reached into the liver. So lastly, it is metabolism. Lastly is metabolism of carbohydrate, or you can call it utilization. Sometimes in exams in Uganda, especially in Uganda, they normally ask utilization. So whenever you are a student from Uganda and you find an exam, especially in allied health exams, they ask you, describe utilization of glucose. They are meaning metabolism. How does the body utilize the glucose? So let us be very quick in it. So this metabolism occurs in two ways. You can call it metabolism, glucose metabolism, or utilization. So once glucose reaches in the liver, you see, if this is the liver cell, this liver cell also has gates. And these gates are called, in the liver it is GLUT2. This is the liver. So before they enter into the liver, high levels of glucose in blood triggers the beta cells of the pancreas to release insulin. We saw the regulatory factors of insulin production is glucose. So once there is an increase in the glucose in the blood, when the glucose is high, it triggers the production of insulin hormone. And this insulin hormone is produced by the beta cells of the pancreas, what we call islets of Langerhans. So when the beta cells of the pancreas release insulin, without insulin, these glucose transporters, they have the gate, they are closed. But once insulin comes, this insulin comes and puts a weight here and leads to the opening of the gate. And then when it opens, glucose is able to enter into the liver cells. So what enables entry of glucose into liver cells is insulin, which leads to the opening of glucose transporters to deliver glucose. That's why we see people with diabetes type 2, which we say they have insulin resistance. That because of not doing exercise, these gates become almost like rusted, that even when insulin comes and sits there, they cannot open. So that's what we call insulin resistance, whereby the liver cells are no longer responding to insulin. So once glucose enters in the liver cells, it is dealt with two ways. Majorly it is the three ways. That the glucose in liver cells, it can undergo what we call glycolysis. Glycolysis, and this is the catabolic reaction, isn't it? It can undergo glycolysis to form pyruvate. Pyruvate plus lactate. Pyruvate, that is in the presence of oxygen, and in the absence of oxygen we form lactate. So glycolysis is the breakdown of glucose to form pyruvate and lactate. But it is always involves also production of energy in form of ATP. And we are saying this is the catabolic reaction. This is catabolic. Why are we calling it a catabolic reaction? In the body, we have two types of metabolism. Two types, whereby one is catabolism, and we have anabolism. Whereby kata is a Greek word which means splitting, isn't it? Or breaking down. Kata means moving down from high to small. Are you getting that? Kata means you are moving from the bigger component to a smaller, breaking it down into smaller components. That's what we mean kata. So catabolism is the breakdown of complex substance into simpler substances, isn't it? And this reaction is hexagonic. The reaction of catabolism, where we are breaking down bigger compounds, we are breaking down bigger substances into smaller substances. We call that this reaction is hexagonic. It is hexagonic because it involves the release of ATP. It is release of ATP. Whereas anabolism comes from the word ana, Greek word ana. Ana means building. Are you seeing that? Building small substances into big substances. That is what we call anabolism. Ana means building up. From small, of course, when you're starting a house, you begin small, then as you go up. So anabolism is building up of small substances into bigger or complex substances, and this reaction is endogonic. Hexagonic means there is surplus of ATP release. That is hexagonic. But endogonic, we are utilizing the ATP without releasing. That is what we call endogonic reaction. Hexagonic, there is release of ATP. Endogonic, you are utilizing. As you are building, you need the energy. That's why we call it endogonic. So we said reaction number one on utilization is glycolysis, whereby this glucose can undergo glycolysis to form pyruvate and lactate, and this pyruvate formed can be fed into Krebs cycle. And this Krebs cycle is the one that provides ATP, water, and carbon dioxide. That is in the Krebs cycle and electron transport chain. When shuttled into electron transport chain and the Krebs cycle, you produce ATP, water, and carbon dioxide as the end product. That's why we said in the functions they are responsible for provision of ATP. Then the second reaction is that in case we have taken much carbohydrates than what the body needs, then there is a need for this excess glucose to be stored. So what the body does is that excess glucose is converted, it undergoes what we call glycogenesis. It undergoes the process known as glycogenesis, and it is converted into glycogen, which is act as a store, which is stored. So that in case there is no carbohydrates we are eating, and there is lack of glucose in the blood, we can break down this glycogen by glycogenolysis to produce glucose, which we can use to provide energy. So we are seeing the excess glucose. This one must be excess. We cannot form glycogen when glucose is not excess. Excess glucose is converted into glycogen. In liver cells and skeletal muscles, this one occurs in liver and skeletal muscles. Then the third reaction, how the body deals with our carbohydrate we take, is that in case the glycogen stores are full, no other place where we can store anymore, this excess glucose which is remaining, what the body does, the surplus, even when the glycogen stores are full, the excess glucose remaining after glycogenesis is converted, or it is converted to lipids. It undergoes the lipogenesis. It undergoes into lipogenesis, whereby we use it, we can convert it into lipids. Excess. Because excess glucose gives us excess acetyl-CoA, and this acetyl-CoA enzyme A can be used for fatty acid synthesis. So that's why even when you are not taking lipids, but you are taking much of the carbohydrate, then what the body needs, you can end up with hyperlipidemia, whereby this excess of glucose can also be converted into fats and stored in adipose tissue. And this is the end of metabolism of carbohydrates. Carbohydrates, that is glycolysis, they can undergo a glycolysis, whereby it is breakdown of glucose into pyruvate. And this pyruvate is can go into Krebs cycle, what we call TCA cycle, and electron transport chain to produce ATP, that is energy, water, and carbohydrates. And lastly, excess of it is converted to glycogen in liver and skeletal muscles. And lastly, when the reserves or the stores of glycogen are full, it can be converted into lipids by the process known as lipogenesis. Thank you so much, guys, for watching this video. Continue to support this by subscribing and putting down your comments. Thanks so much for listening.